morning, student. Today we are going to study regulation of transcription factors. Now we have discussing so far in this session that eukaryotic transcription activators. Eukaryotic transcription activators, they are having DNA binding domain, they are having transcription activation domain. They bind in a specific sequence. There are different module domains are there. With the help of that, they can actually bind to the DNA, and then they are interacting with the basal transcription factor. These are the model we have already studied. Now, how to regulate? Is there any process to regulate these transcription factors? So we can see phosphorylation of activators can allow them to interact with co-activator that in turn stimulate transcription. So phosphorylation may be a key feature. With the help of that, this transcription factor can interact with other protein, which one of them may be known as co-activator, with which in turn help them to stimulate the transcription. Then ubiquitination of transcription factors can mark them for destruction of by proteolysis, stimulation of activity. Now the degradation of a transcription factor, by that also we can regulate the transcription. Understood? The activator, if it is degraded, then we cannot see the enhanced level of the transcription activation. There is another probability that is called summoylation, is the attachment of polypeptide SUMO, which can target for incorporation into compartment of nucleus. These SUMO proteins, actually this is a post-transcriptional modification which change the protein covalently by attaching this SUMO protein, and then can direct where to go. Localization, it may go to cytoplasm or some other compartment inside the nucleus, or it can actually perform many other activity like apoptosis, protein stability, response to stress, progression through the cell cycle. So summoylation has a vast and various activity by which also you can regulate your transcription factor. Then come methylation and acetylation, which we already studied. Heterochromatin formation and chromatin packing euchromatin formation by acetylation, deacetylation can also modulate its activity. When we are discussing this, so a key feature by which these co-activators and activators are working can be demonstrated if we study the signal transduction pathway. This signal transduction pathway we have discussed in your third semester in depth. So we will go just little bit about the signal transduction pathway, and we will see that how it is regulated. Signal transduction pathways begin with a signaling molecule interacting with a receptor on the cell surface. So this signaling molecule, generally it's a hormone, so it's a corresponding receptor which is present inside on the receptor. So you know the nuclear receptor also a type of hormone, steroid hormone, they can also pass through because they are they have this nature, they can go inside, and some other diffusive molecule like NOCO, they can also pass through the plasma membrane barrier. This interaction sends the signal into the cell and frequently leads to altered gene expression. So we understand that when that we have studied that interaction with the signal molecule with this corresponding receptor, there is a conformational change which actually trigger the cascades which all the other proteins and ultimately the gene expression. Many signal transduction pathways rely on protein phosphorylation to pass the signal from one protein to another. We have studied this thing in G protein coupled receptor as well as receptor tyrosine kinase, and so many processes, STAT, JAK, STAT, all the pathway. The phosphorylation is the key feature here. This leads to signal amplification at each step. So each protein getting phosphorylated, its conformational change happen, and it then get activated, and subsequently it activates its downstream protein. Now let's see phosphorylation and activation, a model for activation of CRE linked gene. So you know that G protein coupled receptor signaling pathway, this is a cyclic AMP responsive element we are talking. Now if you see this diagram, what it is showing that there is a Tata box where the basal transcription factors is coming and binding, and you can see there is a CRE sequence, CRE meaning cyclic AMP response element, where the CREB binding protein is coming and binding, but only binding will not actually help it if it is phosphorylated by some upstream protein that you know that who actually phosphorylate CREB. Protein kinase A is there. In terms, it can phosphorylate CREB. Now, the only when it is phosphorylated, then CREB actually interacting with the basal transcription factor with the help of CBP, CREB binding protein, which is also an co-activator here. I told you, and I mentioned another thing, P300 also come here, which is also co-activator and has an HAT activity, histone acetyl transferase activity, if you remember. So that all together actually enhance the transcription. Now, model for activation of nuclear receptor activated gene. Nuclear receptors being polar, it can pass through the plasma membrane. These receptors are actually present where you know that its receptors are present inside the cytoplasm, or in some cases type 1, type 2, it is present inside the nucleus. So what is here happening? This is again the Tata box where the basal transcription factor comes and bind, and this is you are seeing hormone response element HRD, where the nuclear receptor is coming and binding. Nuclear receptor is a protein. Once it's bound with the proper hormone, this hormone and receptor together acts as a transcription factor. That's why we are mentioning. So modification can happen, different type modification. Nuclear receptor binds its ligand, that means the hormone. After binding, it can interact with SRC. You must be knowing response complex protein, which is SRC protein, which is a cytoplasmic tyrosine kinase protein. And this is CBP. CBP is a co-activator and it can interact with the basal transcription factor. Here what you are seeing that nuclear receptor actually interacting with SARC and together coming and interacting with CBP and directly can interact with the basal transcription factor. Here you are seeing that helping is directly with CBP and here helping is through SRC. Now, three pathways that use CREB binding protein and P300 to mediate transcription activation. So these all signal transduction pathways and cross-talking we have studied in your third semester. Okay, so please refresh your memory. So you remember growth factor, stress signal, these are the signals which trigger the receptor tyrosine kinase and subsequently RAS, RAF, all these signaling molecule, phosphorylation, and then MAP kinase is one of the key feature here. MAP kinase get activated. MAP kinase once is activated, it goes in the nucleus and there are so many transcription factor, FOS, June, all these are there. Such like that AP1, SAP1A, these are also transcription factor which comes in turn with the co-activator. Here we are talking of the co-activator. Now co-activator, what is co-activator? CBP-P300. It has an HAT activity and together can go for gene activation. If I go through this pathway, you have definitely studied that steroid, thyroid hormone, retinoids, all the nuclear receptors and its family. Okay, they come inside and they can also interact with CBP-P300 and gene activation. If you remember here, vasopressin adrenaline receptor, these are the hormones. This is G protein coupled receptor, cyclic AMP, protein kinase A, CREB, and then CBP-P300 and then gene activation. All these three way we are seeing that our target is one co-activator CBP as well as P300. So I believe you remember all these things. Now here RAS-RAF signal transduction, what we are showing here, the receptor tyrosine kinase signal induced dimerization and trans-autophosphorylation, which will be subsequently recognized by GRB2 SOS, and then RAS and RAF and RAF then make then ERK. I believe you also can say that the way I'm talking, you also remember maybe more better than me. So ERK phosphorylation go inside and there are so many target. Now here the target change, tissue specificity, signal specificity, the target change. So here they have shown the EIK1, this nuclear protein, this actually helps this transcription factor to activate its target gene. You can write June. Apart from this type of signal transduction, please, okay, one thing I want to mention here, please study the JAK-STAT pathway also. There are other way which we started telling that how you can regulate a activator, transcription activator. One mode is ubiquitinylation. Ubiquitinylation, especially monoubiquitinylation of some activators, can have an activating effect, but polyubiquitinylation mark these same protein for destruction. Generally, ubiquitinylation meaning we are thinking about the protein degradation. If you think like that way, the ubiquitinylation and then followed by the protein is degraded. So that is also another way you are controlling your transcription. Proteins from 19A regulatory particle of proteasome can stimulate transcription. So these are the variety, different types of effect of ubiquitinylation. Now, like ubiquitinylation, activator simulation. What happens if it is simulated? Simulation is the addition of, it's a post-translation addition. That is also, ubiquitin is also post-translation. So simulation is the addition of one or more copies of 101 amino acid polypeptide SUMO. SUMO full form is small ubiquitin related modifier to lysine residue on a protein. Even though it looks like that it's a type of ubiquitin protein, but the function is not like ubiquitin. Most of the cases when we are talking the ubiquitinylation, that time we think that protein degradation, but here it is not a typical degradation. Process is similar to ubiquitinylation, results quite different. What happened? Simulated activators are targeted to a specific nuclear compartment that keeps them stable. So that's one way. So that's why nuclear cytosolic transport, this simulation control the nuclear cytosolic transport of that particular protein, transcriptional regulation, apoptosis, protein stability, response to the stress, and progression through cell cycle. There are so many functions once the cell gets simulated. Apart from this simulation, activator acetylation, non-histone activators are non-histone activators and repressor can be acetylated by HAT. You have studied histone acetyl transferase, so it will actually add the acetyl group to the proteins. HAT is the enzyme histone acetyl transferase which can act on non-histone activators and repressor also. Such acetylation can have either positive or negative effect. If it is a histone, definitely acetylation will cause the more transcription, because acetyl groups are there that will repel each other, and then it will be unpacking, it will be unwounding of the DNA, the chromatin, that region. So these are the way, but that is histone proteins. Here non-histone protein we are talking, because we are discussing what is the fate of the activator by this type of modification. So with this thing, I am ending today's session. Hope this is very easy for you, because you have already studied the entire signal transduction. This is the part of signal transduction where we are showing the transcription factor which are activated. They are actually acting as an activator. So I hope this will be easy for you. Any difficulty, please call me. I will definitely help you there. Thank you for